Hey, Hammy here, coming to you with the first video for AP Biology covering Chapter 8, our chapter on cellular respiration. This chapter is split into 5 sections. In this first video here today, we're going to look at an overview of cell respiration and kind of what the different steps are, redox, and those kind of things, where it takes place. Then it moves into kind of what happens outside the mitochondria, glycolysis. Section 3, what happens if there's no oxygen? We'll cover fermentation. And then moving to the processes inside the mitochondria. And finally, the last video will cover kind of an overview of metabolism. Okay, first off, what is cellular respiration? What is this process? You've heard this before. We've studied it briefly in Biology 1. It's a process that breaks down nutrient molecules. Oftentimes, we're going to say things like sugars, like glucose, produced by the autotrophs, the photosynthesizers, with the concomitant production of ATP. Okay, so we're going to break down a high-energy molecule, get that chemical energy out in the form of ATP that our cells can use for cellular processes, for homeostatic regulation. Cellular respiration consumes oxygen, okay, and it gives off CO2. Okay, the carbon from those organic molecules are going to be given off in the form of carbon dioxide. Because it needs oxygen, we say to do complete cellular respiration, a breakdown of a glucose, it's going to be aerobic. You must have oxygen. Okay. Energy is extracted in steps. It doesn't just take the glucose and because it would just whoosh, it would release so much energy that it breaks it down step by step by step by step in small little steps in order to give out just a little bit of energy at a time. This allows the ATP to be produced more efficiently and not just whoosh, it all lost as heat. Okay, we already lose a lot of it as heat the way it is. And again, we have some, like in photosynthesis, where you had NADP plus as an electron carrier. You have some redox enzymes, NAD plus and FAD, okay, coenzymes that are going to help shuttle electrons in the process of cell respiration. Just like photosynthesis, we want to look at the redox, reduction, oxidation processes that are happening here in cellular respiration, and you'll just see that it's reversed. Okay, so we take glucose and we oxidize it. Okay, we remove those high energy electrons that were put there in photosynthesis, and you're left with CO2. Oxygen is going to pick up those electrons at the end of the electron transport chain and be turned into water. And then we're going to have energy in the form of ATP given off as well. Again, why do we need cellular respiration? Okay, a lot of things that are active processes, cell processes that are active, like active transport, those kind of things take energy. Okay, so we're zooming into the animal cell here, and this happens in plant cells as well. And the cell respiration is going to be, part of it is going to take place and be associated with the mitochondria. Okay, so remember the mitochondria, you have the outer membrane, you have this cristae, these folded membranes inside of here, okay, and then you have the matrix inside of here is the matrix, okay? So you're going to be taking in a sugar, okay, so you're going to have C6H12O6, going to be broken down actually out here in glycolysis, taken into the mitochondria. You're going to have CO2 and water leave along with energy in the form of ATP. You'll also need oxygen going in here as well. Again, the two coenzymes, the electron carriers, electron shuttles that we will talk about, carriers, is NAD+, which is nicotinamide adenine dinucleotide. So NAD+. It's a coenzyme. It helps with oxidation reduction. Okay, it's oxidized when it gives up electrons, and then it'll be reduced. And again, it just shuttles back and forth between the processes and can be used over and over and over again. You can take NAD+, you can add in 2 electrons plus a hydrogen ion. It's going to give you NADH, okay, when it is reduced. Okay? Or we could actually make this a double arrow. When it releases the electrons, when it's oxidized, it will turn back into NAD+. Flavin adenine dinucleotide, FAD, is also a coenzyme of oxidation reduction. Sometimes, some cases, it's used instead of NAD+, and we'll talk about that. It accepts 2 electrons. So notice FAD, it does not have a charge. So if you add 2 negative electrons, you're going to add 2 positive hydrogen ions, okay, because to kind of even out the charges. So that's going to give you FADH2. That's why that has 2 hydrogens. Finally, just kind of an overview of the steps. Sometimes your book will break it into 4 phases or 4 steps for the complete breakdown of glucose. It starts with step 1, just like the name sounds, glyco, which means sugar, lysis, which means chop. Okay, so we're going to take glucose and break it into 2. So sugar splitting, glycolysis. This happens in the cytoplasm. It does form a little bit of ATP, and it is anaerobic. It does not need oxygen. Step 2, the prep reaction before it enters the mitochondria. Okay, the glucose is split into 2 pyruvates. Those pyruvates are oxidized and enter the matrix. You're going to give off some NADH, and you're going to break off 2 carbons, okay, 1 from each of the pyruvates. Okay, so you're going to break off a carbon off of each of these pyruvates up here. Sorry, carbon dioxide. Off of each of these pyruvates up here. Okay, so glucose was 6 carbons, okay, and it's split into 2 3-carbon pyruvates. Okay, down here, the acetyl groups that enter into the matrix are only going to be 2 carbons because CO2 is a carbon here. These are broke off, which are released when you breathe out, okay, when you respire. Step 3, the citric acid cycle occurs in the matrix of the mitochondria. That's like the fluidy inside space of the mitochondria. And we're going to release the rest of the carbons as CO2. And as it does that, it's going to release a lot of electrons when you're, crack. When you're breaking apart the bonds. Okay, remember, bonds are shared electrons between them. Pew, pew, pew. These electrons come flying off. Those electrons, as it is broken down, are going to be picked up by an NADH and FADH2. There is going to be some substrate level ATP production, and we'll talk more about that when we get into these processes. And then the last step is to take all these NADs and FADs to the electron transport chain, where they will dump off their electrons. Okay? And those electrons, again, like a typical electron transport system, the electrons will go boop, 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 boop. And as they lose, they'll pump hydrogens into that intermembrane space. And you have chemiosmosis or chemiosmotic phosphorylation, like we talked about last chapter. That's going to cause chemical gradient, spin an ATP synthase, and phosphorylate ADP into ATP. Okay, again, here's just kind of an overview picture. Notice that they do show a plant and animal cell, fungi. Most organisms on this planet will go through cell respiration. Here's step 1, and actually every living cell will go through glycolysis. Okay, breaking glucose into pyruvate. Notice it's happening out here in the cytoplasm. Okay, and then notice you do get a net gain of 2 ATP for this process. Then it goes to step 2, the preparatory reaction, as it's entering into the mitochondria here, which you're going to give off some more electrons. Glycolysis gave off some electrons. Step 3 happening in here in the matrix. Okay, the citric acid cycle. We're going to, here you broke off some carbons. Okay, broke off 2 CO2s. The other 4 CO2s will break off in the citric acid cycle. So you're going to make a lot of NAD and FAD. All these, see these little electron. All these electrons are coming over here to step 4, to the electron transport chain, or the ETC. That's happening on the Christay membrane, chemiosmotic phosphorylation. Actually, during the Krebs cycle, you're going to substrate level, you're going to directly make 2 ATP. And then the biggie is over here with the chemiosmotic phosphorylation. You'll make 32 or 34 ATP to give you a total of 32 plus 2 plus 2 is 36, or possibly 38 ATP, depending on the organism, and we'll talk more about that and why. So this is just kind of an overview of the mitochondria and the 4 steps and where they're happening and where the electrons are going, the CO2 is coming out, and the ATP being produced.